this podcast, we're going to talk about some examples of non-Mendelian inheritance. We talk about recurrence risk as the likelihood of getting a genetic condition. We've been talking about single gene conditions, whether they be autosomal dominant or autosomal recessive conditions or sex-linked conditions. But what about multifactorial inheritance? How do you deal with recurrence risk? Well, you have to deal with what's called empiric risk. And so, for example, these are just some empiric risks of conditions we know. The prevalence of cleft lip or palate in the general population is 0.001. The same for club foot. Congenital hip dysplasia, maybe 0.002. We just know from empiric risk that first-degree relatives have a 0.04% chance, for example, of having cleft lip or palate. Second-degree relatives, a 0.007 chance. Third-degree relatives, 0.003. So when you're dealing with multifactorial inheritance, you can't really calculate recurrence risk. You have to look at what the data show and just give that information to patients. Some non-Mendelian genetic principles that I think is important for you to know about. The first one relates to mitochondrial genes, and you know this already from your study of biochemistry. Mitochondrial genes are maternally inherited, so you can see the mutation in this individual, this woman, and she's passing the mitochondrial defects through women to subsequent generations. The human mitochondrial DNA, you probably remember this from your study of biochemistry, is relatively small. The mutation rate for mitochondrial DNA is perhaps 10 times higher than that of the mutation rate for nuclear DNA. There are many known mitochondrial diseases. They affect all organ systems. Expression of mitochondrial diseases is variable, and it's variable primarily due to heteroplasmy. This just shows an example of what we mean by that. The disease phenotype is variable as more and more mitochondrial mutations accumulate in an egg. Offspring can show effects from no effects to relatively mild effects to relatively severely affected. Now I want to talk about gene silencing, because for some human genes, one allele may be transcriptionally inactive. That is, there's no messenger RNA that's encoded by that gene, and that's dependent upon the parent from which the allele was received. And this is something that we've discovered only relatively recently. Gene silencing, we would say an allele is transmitted by the mother, would be inactive. Maybe the same allele transmitted by the father would be active. So in gene silencing, a normal child would have only one transcriptionally active copy of a gene. Under most circumstances, you have two transcriptionally active copies of the gene. So the process where one gene is silenced is known as imprinting. The transcriptionally silenced gene is said to be imprinted, and we know now that there are several dozen human genes that are imprinted. Imprinted genes tend to be heavily methylated, and the histones associated with these imprinted genes tend to be hypoacetylated. So imprinting of genes on the autosomes is in a sense similar to X chromosome inactivation. We know that imprinting appears to be a normal process, but imprinting can cause problems, because what would happen if there's a chromosomal aberration, say a chromosomal deletion, in a region that involved imprinted genes? There's a 4 megabase pair region on chromosome 15 on the long arm that, if it's deleted, can cause problems. If the deletion is inherited from the father, the child is going to have a disease known as Prader-Willi syndrome. The child will have short stature, poor muscle tone, relatively small hands and feet, will be obese, will have some mild intellectual impairment, and will show hypogonadism. That's Prader-Willi syndrome. If the same deletion, instead of occurring on the paternal chromosome 15, occurs on the maternal chromosome chromosome 15, the child has Angelman syndrome. In this case, the child has severe intellectual retardation or intellectual impairment, seizures, and ataxic gait. The diagram on the bottom explains how this occurs. So we know that on chromosome 15, there's regions that have active Prader-Willi genes and inactive Angelman genes. It turns out that on chromosome 15, the paternal chromosome 15 has the Angelman genes silenced, and on the maternal chromosome 15, the Prader-Willi genes are silenced. So here's what happens. You have a deletion on the paternal chromosome 15. You lose the active Prader-Willi genes, and the individual child has Prader-Willi syndrome. If the deletion occurs on the maternal chromosome 15, you are deleting the active Angelman gene. The child still has the active Prader-Willi gene, so the child has Angelman syndrome. If the single active copy of these genes is lost through a chromosome deletion, there's no active gene product, so the disease results. Another situation we've come to understand is anticipation, that is, where there are nucleotide repeats. Myotonic dystrophy, it's an autosomal dominant disorder. It can appear earlier and earlier in each subsequent generation. The disease is caused by a trinucleotide repeat expansion. It's a CGT repeat in a protein kinase gene, and this repeat expands in subsequent generations. So the disease is manifest earlier and earlier in each subsequent generation. The southern blot on the right shows a homozygous individual, normal 4 to 5 repeats. A heterozygous individual may show 175 repeats of the disease gene with this trinucleotide repeat. Here's a heterozygous individual several generations later that shows 900 repeats of the trinucleotide repeat. So in this instance, the disease would manifest at a younger age than in this instance, for example. There are a variety of diseases that are associated with nucleotide repeat expansions. Huntington disease is a trinucleotide repeat expansion in a CAG repeat. Normal individuals show 6 to 34 or so repeats. If you show above, say, 35 repeats, that suggests that you have the disease phenotype. The more repeats you have, the more severe the disease, and the earlier the onset of disease. Huntington disease, as you know, is an autosomal dominant condition, loss of motor control. You'll have dementia eventually coming into play, and a very much an affective disorder. Friedreich ataxia, another nucleotide repeat syndrome. This one's an autosomal recessive. 6 to 32 repeats in a GAA repeat, that's normal. 200 to 1700 repeats suggest you have the mutation and the disease manifest. Has limb ataxia, disordered speech, motor functions. There may be cardiomyopathy. There can be weaknesses in the lower extremities. Fragile X, another trinucleotide repeat in a CGG repeat. This is an X-linked recessive disorder. 4 to 39 repeats is normal. You get above 200 repeats and you begin to show the disease phenotype. Mental disability, large ears and jaws, maybe microorchidism in males. And we know there are several dozen diseases now that relate to nucleotide expansions. So there are conditions and genetic diseases that you may encounter that do not necessarily follow simple Mendelian patterns of inheritance. We've talked about osteogenesis imperfecta in relation to germline mosaicism. We've talked about retinoblastoma, where there's incomplete penetrance. We've talked about Huntington disease, trinucleotide repeat, but an age-dependent penetrance. Variable expression, we mentioned neurofibromatosis. Locus heterogeneity, like adult polycystic kidney disease. Some diseases show pleiotrophy, where there's more than one effect. We mentioned, for example, Marfan syndrome, cystic fibrosis. We've mentioned sex-linked diseases like hemophilia or Duchenne's muscular dystrophy. We haven't mentioned mitochondrial diseases, but a classic one would be Leber's optic neuropathy, and there are many, many mitochondrial diseases. We talked about Prader-Willi-Engelmann syndrome for genomic imprinting. Beckwith-Wiedemann is another example of a disease showing imprinting. And of course the nucleotide repeat expansions like Huntington disease, Fragile X, and maybe 20 or so more diseases related to nucleotide repeat expansions.